This is TWIS. This Week in Science, episode number 927, recorded on Wednesday, May 24th, 2023. How did we get here? I might not have all the answers, but I am Dr. Kiki, and thank you for joining us for another evening of This Week in Science. Tonight, we will fill your heads with spiders, Saturn, and solar flares. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! Some days you just pick your head up and look around and wonder to yourself how you got where you are. Is this the place you meant to be? Really? And does it really matter? You are here. Well, choices, luck, ignorance, knowledge, there's lots of ingredients that go into that magic soup. Sometimes it involves solar flares, and sometimes it is just This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
Oh, and a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are so glad that you all joined us for this episode of science. You know, we like to talk about science every week, and I hope that you like to be a part of that. Thank you for being here. Thank you. I hope you're doing well. All right, today, what do we have? I have science news stories about life, vibrations for brains, and Saturn. What do you have for us, Blair, in the animal corner?
I have spiders. I have more spiders. I have frog sperm, and I also have, what was the last one? Oh yeah, dinosaur eyes and bird brains. It's a whole thing.
I thought you were going to say more spiders.
No, just the 2 spiders this week. Just 2. Hopefully that's enough.
Only 2 spiders, that's all. It's enough for Blair. It's enough for me. But if you're all excited about these stories and the other ones that we didn't even mention, because of course there's going to be more that comes up. You never know what's going to happen here. I would love to remind you before we jump into the show that you can subscribe to TWIS, all places that podcasts are found, and that we broadcast on YouTube, Facebook, and Twitch weekly on Wednesdays at 8 p.m. Pacific time. And additionally, you can find us on social media. Look for This Week in Science or at TWIS Science. We're at TWIS Science on Twitch also, by the way. But if you want show notes and audio files and all that kind of stuff, you can find all sorts of information at our website, twis.org. Okay, let's jump on in. What would the Earth be without the sun?
Cold and dead?
Pretty much.
Okay.
And not just the cold part, but the dead part, especially. Yeah. New research that was published this last week in the journal Life. An article called Formation of Amino Acids and Carboxylic Acids in Weakly Reducing Planetary Atmospheres by Solar Energetic Particles from the Young Sun was published, and the basic gist of the whole study is that solar flares and really energetic solar flares were really important to sparking life on the early Earth, which means that additionally maybe Mars was another spot where life was started, because we know from lots of experiments, and we've talked many, many times about early life experiments where just from electrical stimulation and a few basic ingredients, we've been able to start to form amino acids and some of the basic building blocks of life. And we've found building blocks of life all over the place. And it just gets, the story has gotten more and more and more concrete that life should be starting everywhere. But what is the thing that really starts it, right? What really gets it going? And this particular study really took the age of the sun into account and the activity of the sun into account. And so nowadays we might be afraid of the occasional solar storm and the possibility of solar flares that could, electromagnetically wipe out our electrical grid or affect our satellites for communication. But once upon a time, our sun was a lot more active. It was like a hyperactive little tantrumy child.
Sure.
Yes. And because of that, the interactions of the super flares that we've really never even encountered during our time here on Earth, or what we've been existing in recently, that this cosmic radiation from our sun would have sparked reactions within the atmosphere and within various components of the Earth itself to create plasma and to create amino acids, carboxylic acids, and would have been better than just lightning on its own or galactic cosmic rays. So just our sun by itself, being a tantrumy youngster, might have led to the biomolecules that made life possible.
I love that. I think everything we know about thermodynamics tells us that energy is important and that it fuels a lot of things in life and the universe, and that we know that a lot of chemical reactions that it takes to make life happen require energy input, right? So I guess, yeah, that makes sense to me. It follows, right?
It follows, but I think the one thing that is kind of shocking to people thinking about how the sun might have been involved in it is that the sun used to be, when the Earth was very young, a lot dimmer, about 30% dimmer. But even though it was very dim, it had these massive eruptions.
Right.
And it was the energetic particles that would come and the electromagnetic storm that would come as a result of these super flares that were probably at least once a week. Not like, once every several years, once a week.
Yeah.
That
Well, and I guess that also helps to explain why life isn't happening literally everywhere, that there is a star that's a good distance, right? So if you need this kind of secret ingredient that is the secret spice of the solar flare, then you need it at the right distance and the right heat and the right intervals and like all these specific Goldilocks-y things that make life happen. So that helps explain why it's not happening everywhere all the time, I guess.
Not everywhere all the time, and why like our sun isn't destroying us constantly now.
Thank you. Thank you, soul.
But it also does give us that perspective on other planetary systems, other solar systems that might be younger than ours, and what might be happening there, and those that are older than ours, or that life cycle. We now have maybe something that's a little bit gives us some more ideas that we can use for comparison.
And gives us places to look. Where are there stars that are having periodic solar flares close together that might help spark life? Let's go look.
Let's go look for all of those stormy solar flares.
Look at that go. Oh my gosh.
Life.
That's a firework if I've ever seen one.
That's right. Thank you, sun. Thank you for being quieter now. Thank you for being very active once upon a time.
Yes.
What did you want to talk about next?
Oh, okay. Let's talk about spiders and
right into the spiders. Okay.
Yeah. Well, I have an interesting angle on this study. So this is a study on funnel web spiders, which Sydney funnel web spiders are extremely venomous. They're super dangerous. The development of antivenom is the main focus on this species. So almost all research done on funnel web spiders is looking at antivenom, which is good. I'm not complaining about that at all, because if I ever go to Australia, there are so many things that could kill me, and I appreciate antivenoms for as many as possible. So, that being said, though, they're not being studied for much else.
Oh, wow.
And so there's a lot of kind of gaps in our knowledge of funnel web spiders. So, the other issue is that funnel web spiders, as the name suggests, live in funnels. And so it's also kind of obscured and difficult to study them naturally in the wild. So this
because
is
they live at the bottom of a funnel.
Yes, exactly.
They come out to attack things.
Yes, precisely. So this was a study looking at courtship behavior and mating systems in the Sydney funnel web spider. And there were a lot of kind of guesses based on the anatomy of these animals. And they found some things they were expecting and some things they weren't. So they knew that there's kind of a display that happens outside of a female burrow, because again, that's something that you can watch pretty easily in the wild. Videos have been collected a lot to describe the male and the female kind of behaviors that happen outside. And then now they've collected videos for what's happening inside their burrows in a lab. So they had mating pairs filmed in 451 videos to create 165 minutes of footage in total. So they were able to really see what these guys were up to during courtship and mating. And so after they have this kind of elaborate display and the female welcomes them in or concedes or whatever you want to call it, they found some kind of lifting behavior, which is really interesting. And in the end, some of the males would be chased away. And so based on that information, the initial guess was that the males were lifting up the females to keep them from eating them. So basically, so the female could not attack, kill, or eat the males, which is a very common story in spider mating, right? So they're basically like, oh, can't get me, can't get me, can't get me. So they have these clasping spurs. The males have these clasping spurs on their second pair of legs that is used for lifting the females. So after watching these videos initially and seeing that a couple ran away, they're like, oh, that's what it's for. But as they continued to study their videos, they found that the spurs are actually used to pull the female toward the male and keep them there. It's just a handle. It's really just a handle. And this is really just a selective function. And they didn't see any cannibalism happening between these spiders or any reason to believe that that might be happening in the wild. So,
so these very venomous spiders that are very like dangerous spiders to humans, they're not attacking each other. They're like, hey, we're together.
Come into my funnel.
Come on in.
Yeah.
I invite
then you
in.
can leave though. When we're done, you can leave. Anyway,
I'm only going to hold on to you while you're here.
Yeah. But so this is interesting because we're looking at an animal that's difficult to study in the wild. There are a lot of guesses made on the fact that these are a dangerous spider to humans. I think that definitely colored the conversation a little bit. And that coupled with the fact that a lot of spiders are cannibalistic in mating. So they kind of put two and two together with these spurs and kind of made this guess, which there's nothing wrong with that. That's how a lot of zoology started, is observational based on characteristics. That's how originally animals were lumped together before we learned about genetics. So all this kind of stuff, right? So that's a place to start. But I think this is a great example of using that kind of evidence to make a hypothesis and then be able to test it in a lab and be able to actually see, oh, nope, it's used for something else entirely. I totally understand why they made that guess, but I like that these extremely venomous, scary, dangerous spiders are actually not so bad to each other.
And from what it looks like, that maybe the attacking of mates isn't, it's not widespread, and maybe it's simply just, evolution moving a particular direction for different species, right? And that in this particular case, you have two similarly sized, very, venomous creatures that maybe they don't need to do that. Maybe because of the size, or there might be all sorts of reasons why it doesn't work out in that direction.
Yeah.
But their hypothesis was wrong.
Yeah. They're not so bad, the funnel web spiders, unless you're a human or any other animal being bitten by them. Then seek some anti-venom, please. Go to the hospital.
Seek medical help as soon as possible.
Yeah. Stay away from funnels in Australia. That would be my suggestion.
These are some of the most venomous spiders in the world, right?
Yeah.
Most dangerous. Yeah.
Mm-hmm.
Yeah. Oh, awesome.
So they brought a bunch into a lab. What could go wrong?
I am inspired by those scientists who wanted to work with these spiders.
Oh, man. That's wild. It's like, bye, dear. Hope to see you at the end of the day. Unless I accidentally get bit by a funnel web, then see you never.
I'm sure they have anti-venom.
Oh, yes.
Available. I'm sure they've got something. Yeah. Oh my goodness. Well, something that's equally deadly, but not something that you find at the bottom of a funnel or a web. Saturn. I mean, it's deadly to us. We can't go there ourselves.
What are you afraid of?
There's no anti-venom for Saturn. Nope, not happening. Okay. So this week, a couple of studies came out that were related to Saturn that I thought were very exciting. Researchers talked about their work with the James Webb Space Telescope looking at Enceladus, which we know has plumes of what we think is water, vapor, and James Webb's telescope, according to researchers that reported at a conference at the Space Telescope Science Institute in Baltimore on May 17th, that there's massive plumes, and James Webb saw them all. So this isn't the first time, but this is like the highest resolution that we've been able to see these plumes with. And the jets of vapor, they say, shoot further into space than they previously realized, further than the diameter of Enceladus itself. So there's so much pressure. It's just spewing this vapor out into space. Their material is part of the forming of one of Saturn's rings. So these vapory blasts causing ice particles actually lead to the formation of one of the rings of Saturn, which is also very exciting. Analysis suggests that the jets contain methane, carbon dioxide, and ammonia, which are all organic molecules. These are necessary for life. One of the hypotheses is that beneath the icy surface of Enceladus, there may be oceans of methane and these organic liquid molecules that could be harboring bacterial life. But we don't know that for sure because we have to go there and we have to see that for sure. But it's a very exciting idea. And now maybe there's more evidence from the James Webb Space Telescope that will push us into exploring Enceladus even further.
You got any pretty pictures of that? Blasting that you're talking about?
You got a pretty picture. Well, the pictures from the James Webb still need to be analyzed, and the paper needs to come out. But there are, yes, some gorgeous pictures of these jets shooting out from the surface of Enceladus. Yeah. They're
part of that early reporting from NASA we were talking about before. Come on, don't tell me what the James Webb saw without some pictures.
Give me the pictures first. I want to see them.
You're a camera, right? Isn't that your whole deal? Give me some pics.
It's the whole deal. Additionally, not related to the James Webb Space Telescope, but we all know the Cassini mission that spent years studying the rings of Saturn, studying Saturn itself, and taking just some beautiful, beautiful looks at the rings and this incredible planet. Well, we kind of take these rings for granted, right? It's these rings of Saturn. We look at them and go, wow, it must have been there. And they've formed over such massive time periods. And we think of our solar system as this kind of stable thing. But new research that has been published this last week, based on some of the last data available from NASA's Cassini mission, suggests, based on theoretical models, that these rings are really only a few 100 million years old. So younger than the Earth, younger than the moon. Probably were getting their start sometime in the time of the dinosaurs.
Okay, so let me ask this question. If I was on a planet far enough away and I had a telescope where I could see Saturn, I would see it without rings.
Yes. Yep. Yes.
Yep.
Exactly.
Very cool. Or if I had a spaceship that traveled fast enough, I could look back.
You could look back
and see no rings.
Yes. Right?
That's wild.
It's really wild. And other ideas that come from this data and this work suggests that maybe the other massive gas planets in our solar system, which also have rings, by the way, we just can't see them very well, that maybe their rings were bigger and more prominent at one point in time before we started really looking.
Sure. So they're like, I wore rings before it was cool.
I wore rings. I just thought, I've been getting rid of them. It's okay. It's fine. So the models, based on what they got from the Cassini data, it demonstrates that the rings are actually losing mass onto the planet. So that there's, we talk about meteors hitting our planet every once in a while, but basically those rings are just like being pulled in by Saturn's massive gravitational pull, and there's meteorites constantly falling in from the rings, and the rate of stuff that's falling suggests that they're really only going to be around for a few 100 million years from now. So well before our sun decides to expand and destroy us all, Saturn will have rings that we can't really see anymore. Which is so fascinating.
We're so puny and small. We mean nothing. Just think, we're such a little blip, and even our own solar system has such a history on either side of us. It's wild.
Right? That someday, we're just lucky to be seeing what we're seeing at all.
Yeah.
Yep. How'd we get here? Thanks, solar flares. Who knows what's going to happen next? I don't know. We don't know. We'll see where it goes. Do you want to tell me about frog sperm?
Yeah, of course.
Does it sound like a very natural segue from rings of Saturn?
The rings of Saturn to frog sperm? It's all eternal. Anyway, no, this is a real quick story that just it's from University of Newcastle, and it's a breakthrough in cryopreservation of amphibian sperm, which I think everybody listening is acutely aware at this point that amphibians are not doing great. They are very sensitive to changes in water quality and changes in temperature and all sorts, new fungus introduced to the water and all sorts of things. Amphibians are called indicator species because they are so sensitive that they're a useful tool when kind of checking on the health of an environment. They're usually one of the first things to be impacted. But so because of that, there's a lot of conservation efforts to raise amphibians in a controlled setting and then release them when they're adults into the wild so they have a head start. A lot of programs are working on inoculating them against chytrid fungus and other things in the environment so they have a better chance of survival. And remember, they also have important ecosystem services because what do they eat? Bugs like mosquitoes. So all that to say,
yay, frogs.
This is why we care about amphibian sperm, because there's breeding efforts that happen in captivity to help continue these amphibian populations that are in trouble. But I wasn't aware of this. The methods are imperfect. And I think about like human fertility and how eggs can be frozen and sperm can be frozen and all these things. And it sounds like it all just works. And I just kind of had the assumption that it works and we got it and we figured it out and we can freeze sperm and it's not a big deal. But this recent research found a superior method of freezing and thawing frog sperm, which had improved sperm quality as a result. They found that using less sucrose or sugar in the freezing process, which who knew sugar was involved, but it is, facilitates higher cell recovery, resulting in more intact membranes and better sperm motility, which produces, of course, better swimmers, which produces more viable offspring. So they tested sperm from 6 frog species. In all cases, the new preferred thawing method was superior than the traditional cryopreservation method. So pretty straightforward study. They just tried a new way to freeze sperm. It worked really well. This could have a huge impact for amphibian conservation. So good job.
It's kind of interesting, specifically considering that they're amphibians and they lay their eggs, then they release their sperm usually in a watery environment. So perhaps that's some part of why the difference in freezing, like less sugar, makes the difference.
Yes. I would imagine that a pond is very different from a reproductive canal in terms of many things. pH, sugar, lots of other things.
Yep.
So,
At least I hope so.
It's very different.
But yes, sucrose is used very often for cell preservation, for sperm preservation, because when you freeze stuff, if you don't do it in the right way, and they use sugar very often because what it does is it creates a different diffusion gradient so that the cells, as they freeze, the water molecules don't get spiky and make ice shards that shatter the membranes.
That makes sense. Oh man, that totally makes sense. Which also, since you brought up the fact that a lot of them are broadcast spawners and they're just spawning generally in the environment, also some of them live in ponds that freeze. So
so
they're
maybe
probably more tolerant.
Their cells might be more tolerant.
Yeah. That's really interesting.
Yeah.
Think about that.
And mixing it up with the sugar is probably mixing up the metabolism of the cells, messing with actually how the molecules are working. And interesting. But I'd love to see how this translates to other amphibious species and maybe either other cold water dwelling species, fish, and I wonder. I don't know. Speaking of cold, the Montreal Protocol has been doing its job. Do you know what the Montreal Protocol is, Blair?
No, I don't recall. It sounds familiar.
Yes. Well, we like to think of it as the agreement or global treaty that decided everyone around the world basically decided to reduce ozone that we put into products, or the CFCs that we put into products that cause ozone. So it was the ozone protecting protocol. And according to, this was about 1987 when the treaty was signed. And you hear every year, the ozone hole, it's opening or it's closing, it's getting smaller. And we think of it in terms of sunburns and cancer. But it also has climate impacts because the ozone layer is part of the atmosphere, and a new study that was led by researchers at Columbia Engineering, the University of Exeter, has shown that the treaty has slowed down the effects of climate change on sea ice in the Arctic by as much as 15 years.
That's great.
The appearance of the first ice-free Arctic summer it would have been earlier, but we got rid of those ozone causing pollutants in the atmosphere. At least not got rid of, but we minimized it. And that was something we did together. Globally. So in terms of solutions, communications, this, I think, is one of those stories. A global community not a global community, the global community came together, made an agreement. We stuck to it, and we have had an impact. We can do more.
Yeah, I think about the CFC ban all the time in relation to kind of how much traction was made over such a short amount of time, versus how slow kind of the fossil fuel phase out is going. And it's proof that we can do it for sure. But I think it's a lot of it was something that we discovered quickly, wasn't being used in that many things. They were being used in very specific items that there are replacements for. Very easy, not that fiscally impactful replacements for the CFCs. Nobody really noticed when you stopped putting it in hairspray, for example. Yeah. There are other aerosols you can use. And so I think that it was a pretty easy switch and replace. Not to say that it's not important. It's a very important thing that happened, and it proves that we can do these sorts of things, but it definitely was a lot less complicated than trying to unentangle ourselves from fossil fuels, which is in everything.
That's a much bigger task for sure. Yeah. But I think it's these kinds of agreements where it's evidence that the global community can come together and do things that are successful and have impacts. And so we need to remember that. It's a big task, but bit by bit. Yeah. Little bit by little bit.
Well, and we're making huge strides. It's happening. Yeah. I mean, I went to a webinar a couple weeks ago of like, let's talk about what's actually working in climate change. And I was really surprised. You don't hear the success stories, but what was it? Electric car purchases are up 10 times in the last 5 years, I think. 1 in every 7 cars bought currently is an electric car. Yes.
Amazing.
You don't hear these things, right?
No.
Like huge numbers of houses are being built with heat pumps instead of air conditioners, which work just as well, if not better, and don't use fossil fuels. Like there's all of these things that are happening that we don't talk about, and we don't stop to celebrate successes, even though that's what can encourage further change. So, yeah.
Let's celebrate the successes. I think that will help us remember that we have the power. Like,
you hear that, ozone layer?
Kira.
We saved you.
We saved you. We're going to save more things.
Yeah.
We'll do it. I don't know. We're helping wombats? No. What's the New Zealand species that we've been helping cure their cancer? But anyway.
Tasmanian devils.
Tasmanian devils. We've been helping with that. We can do that. We've been working on things.
Yeah.
We fix stuff. It's good. This is This Week in Science. Thank you for joining us for another fun-filled episode. We hope you're enjoying the show. And if you are, please share it with a friend. Right now. Text a friend, call a friend, be like, look at this show. You should watch it. You should listen to it. Subscribe. Do that. That'd be very, very helpful. And if you really love the show and you want to help us do what we do every week, head over to TWIS.org, click on our Patreon link, and become a supporter. Every month, you can support us at the amount of your choosing. $10 and more, we will thank you by name at the end of the show. $15 and more, you'll get a sticker every once in a while. There's stickers. Come on, everyone. Help us out. We can't do this without you. Thank you for your support. And now that I've thanked you and we're coming back into the show, I think it's time for Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have a study about dinosaurs. Or
Dead
more
animals.
Specifically, it's actually about alligators and ostriches. But researchers have decided this study on alligators and ostriches tell us about dinosaurs.
Okay.
So it's, in my opinion, I don't want to color people's opinion before this even starts. It feels like a little bit of a far walk, but we're going to take this walk together. And we can discuss if this sounds correct.
We will
So,
discuss.
a study from Lund University in Sweden, they were looking at visual perspective taking in animals. So that's a skill that usually emerges in humans around age 2. But this is the idea when someone near you turns their head towards something in their environment, you follow their gaze. But specifically, the more advanced behavior is when you follow someone's gaze to a location that is initially obstructed. So, like, if I'm in this room and Brian's outside the door and he looks towards our front door, then I would walk around the corner and look at the front door. I would be able to understand he is looking in that direction. Let me go see what he's looking at. That is visual perspective taking. So that means I can put myself in his shoes and figure out what he's looking at, right? So, and that's without having to like do a bunch of double checking over and over and over of what's your deal? Oh, let me stand exactly where you are and look at what you're looking at. No, I can understand based on where I am and where you are what you're looking at. So that's visual perspective taking. This has been observed mostly in mammals, some birds and reptiles, and this kind of very specific tracking of where a person's looking is mostly attributed to apes, monkeys, and ravens specifically. So they wanted to see when this started happening, because as this stuff often does, it was attributed to humans and other apes. It's a very sophisticated thing that's happening. And so the more you look, you see other animals that do this. And so they wanted to see when this behavior emerged. So they used a comparison of alligators with the most primitive existing birds, paleognaths. Those are ostriches, emus, rheas, and one of my favorite types of birds, tinamous, which I will suggest everyone looks it up. Tinamous, they're little cartoon birds. There's really no other way to describe it. They look like little loafs with bird heads. Anyway, They run around on the ground. They are technically flighted, but they're flighted like a chicken is flighted. They're not super great at flying, but they are flighted as compared to ostriches, emus, and rheas, which don't even have flight muscles at all. So they wanted to see the neuroanatomy, or specifically, they looked at neuroanatomy of these two groups because they're pretty similar. So basically the idea is crocodilians and birds and their common ancestor, which was a dinosaur, they all kind of have the same brain because their neuroanatomy is similar. So if you see similar behaviors in these individuals, chances are they're based in similar neuroanatomy, which means they're based. Yes. So it's already, you see where I'm getting with this is like a little bit of a leap. But so they're comparing ostriches, emus, and rheas, these paleognaths, to their forebears that had similar brains, which are velociraptors and similar. Okay. So they wanted to compare these two groups, which creates a bracket around an extinct lineage of dinosaurs, which leads up to modern birds. So basically, either they share this behavior and a common ancestor had it, or they do not share this behavior, and therefore a common ancestor did not have it. So here's where, in my opinion, this study kind of takes a turn. So
evolutionary psychology behavior stuff is always a bit tricky, but
you can't test it. So it's anyway. So the alligators didn't demonstrate visual perspective taking in their tests.
Okay, so they're a little out group, little further removed from the velociraptor group from which birds are derived.
Right.
Yeah.
Every bird species they test exhibited visual perspective taking. Also, these birds engaged in a behavior called checking back, which is where when they look somewhere, then they look back at the individual's eyes and then retract the gaze, kind of understand, like, I'm looking at what he's looking at, right? And previously, that's only been seen in humans, apes, monkeys, and ravens. And so, based on all this information, When paleonath birds emerged 110 million years ago, they're saying that that's when this behavior emerged. And so that means it predates primates and dogs who have been doing this behavior by 60 million years. Looking at the anatomical similarities between these birds and their non-avian forebears, they say, quote, "It is plausible that the skill originated even earlier in dinosaur lineage. However, it is less likely to have been present among the earliest dinosaurs, which had more alligator-like brains." So this is where this study loses me, because just because ostriches, rheas, and tinamous all can do this does not mean that their common ancestor definitely did it.
No. It's a behavior.
It's a behavior.
Yeah. I mean, so I get what they're getting at, but
you can't test it. But also, my dear researchers, what about convergent evolution? This does not have to be a common trait that dates back to dinosaurs, because mammals have this and birds have this. Therefore, the common ancestor with mammals and birds, but not with crocodiles, have this. That is not necessarily the case. Like, all these animals are social. All these animals require interaction with other individuals. This could be something that has popped up a million times. They all have eyeballs. The eyeballs are the convergent, are the common evolutionary trait.
Yeah.
They all have vision. These animals all have vision. So, like, they have vision and they have a social structure. Therefore, at some point, it's evolutionarily beneficial to be able to pay attention to what my friend's looking at. In no way, to me, does this mean that this is something that was evolutionarily conserved for 110 million years.
I think what you're getting at with the sociality is the most important aspect of that. Is like, once you are cooperating or working with other individuals, you need to know what they're looking at. Right. There's even a certain aspect of predation that, okay, maybe the alligators they tested, that wasn't the group, but looking at what other species in your ecosystem are looking at and knowing when there's a threat, when there's not a threat, maybe it's, I don't know. There's a lot of stuff in there that is not necessarily being taken into account.
And that's exactly it, right? Crocodilians are, they're not pack hunters, and they're ambush predators. They can climb trees, as we've learned, but they're ambush predators. So they don't care what anyone else is looking at. They're waiting for their food to practically touch their nose.
Right.
But wolves, where dogs come from, are pack hunters who hunt by sight and smell. And so not only do they cooperate in their hunting, but they need to be able to track moving prey.
Yeah.
All of the birds that we talked about are lunch. So it's really important for them to be able to see if somebody else noticed a predator.
Exactly.
So, like, the crocodilians are also just a really weird choice because they have such a different behavior set. Anyway, this was all over the internet. I saw this posted maybe 6 times this week. Dinosaurs are better than dogs at tracking. It's understanding perspectives.
We have no idea. This is an interesting study. It's a great comparison. Love their approach, but we don't know. Yeah, and maybe some dinosaurs, yes. Other dinosaurs, no. I love convergent evolution, adaptation based on need.
Yeah, it's.
I don't know.
Did velociraptors do this? Maybe.
Probably.
They did, to our knowledge, pack hunt, right? So, sounds good.
Probably.
Did T-Rex do it? Probably not, because they were largely scavengers. So their stuff isn't moving; it's dead already. I will.
And they were big. There wasn't a lot for them to worry about.
Yeah, exactly.
Get out of the way.
I do have a quote from the senior author, Professor Matthias Osvath. It's a really good quote, which I agree with mostly. Early in my career, crow birds earned the nickname feathered apes due to numerous research findings that showcase their remarkable cognition. However, I'm beginning to question whether it would be more fitting to consider primates as honorary birds.
I like that. Are you a bird? Well, I mean, really, we're all reptiles. So
Yes.
that's where it goes back to.
I mean, we're all fish if you want to go that far back. So
Truth.
it's a whole thing.
It is.
Anyway, when is an ant a plant? And when is an ant that is a plant actually a spider?
What? Do tell.
I think we've talked before on the show about jumping spiders that look like ants.
I think so.
Yeah, so they like stick up their front legs so they look like antenna, and they walk in a very particular way to try to fool potential predators into thinking they're ants. Mimicking ants is a great defense because usually they're not very delicious. They have spiny defenses, they bite, and a lot of them carry chemical repellents or venom. So if this little jumping spider wants to avoid being eaten by other spiders, looking like an ant can be a really good way to get away with it. But this particular jumping spider, Scylla collingwoodi, was already known to move like an ant and kind of look like an ant. But one thing that was weird about it is it was covered in these really bright colors. And ants aren't these colors. They're like red and orange and white and black.
They're not pretty abstract pictures.
No.
Usually. No. They're
No.
solid. Ants are usually just a solid color, maybe a couple of colors.
Why are you moving like an ant and looking like an ant, but colored like I don't know what? So they wanted to do some studies to see who they were trying to fool and how with this stuff. From a human's perspective, it looks like they blend well with the plants that are in their environment, but they wanted to test that to see if it actually helped with predators. So they collected wild ant mimicking spiders from 4 different locations in China and brought them back to the lab. And for comparison, they collected another kind of jumping spider that doesn't mimic ants and 5 co-occurring ant species so that they could kind of see like who did they blend it with. And in the lab, they characterized and compared how the ants and spiders moved in terms of their limbs and their speed and their acceleration and whether they followed a straight path or they took something kind of back and forth. And they found that jumping spiders usually jump. But these jumping spiders, they didn't. They moved like the ants. They actually did appear to move very ant-like. They bobbed their abdomens. They put their front legs up to look like antenna. They lifted their legs to walk in an ant-like manner. So they even like the way they moved their legs were similar to how ants move. They most closely resembled the smaller ant species that are closest to them in size, because jumping spiders are teeny tiny.
Little tiny.
Yeah. So it seems like a really effective one. But they weren't a perfect mimic to any one species. It seemed like they kind of got mostly there to 3 different species. And so you can see how that could be beneficial, because being a general mimic rather than perfectly mimicking one ant species makes it more possible that they could expand their range, that they could go in different habitats, that they could go into different types of environments and still blend in with the other ants where they live. So then they tested their the spider's defenses against 2 of their predators, a similar sized jumping spider with color vision that does eat other jumping spiders, and a praying mantis.
That will eat jumping spiders and ants and anything it can get its little mandibles
They'll
on.
eat anything.
Yes.
The other difference is that jumping spiders, as you might guess, because they're always so colorful and beautiful, usually, they have color vision. And as far as we know, praying mantises have a monochromatic visual system. So that is where this kind of weird coloration comes into play. So they looked at how the predators would see these spiders relative to other prey species, and they put them against plants as well that the spiders would live on, which include red flowering West Indian jasmine and Fukian tea tree. And they found that the ant mimicking spiders were better camouflaged for both spider and mantis on the jasmine plant. So they do appear to blend in well with a very particular plant where they live.
Mm-hmm.
But when they were just given us the option to take a spider and eat it, and they were like, "Oh, is it an ant? Is it not an ant?" Looks like the predatory spider would attack the non-mimic jumping spider. Out of 17 trials, they launched 5 attacks, all on the non-mimic. But praying mantises, they ate everything. The idea there is that praying mantises, as you kind of alluded to, they're huge and they can eat spiny ants. Doesn't bother them.
Right.
So, of course, the mimic wouldn't really work for them. But the predatory spiders would get injured by a spiky ant. They would avoid those. So it does seem like it's a targeted mimic towards other spiders. They're doing their best. They're blending in with a specific plant, and they are attempting to mimic multiple ant species, which is very cool.
So it's a good enough mimicry.
Yes.
It's a mimicry that's kind of general, but able to blend in well enough to not die most of the time.
It's evolutionarily beneficial, right?
Yeah.
So, like, again, this isn't the end of the evolutionary line. I don't know how many millions of years it's taken them to get here, but in another couple 100,000, maybe they'll be better at it, or maybe they'll add something to it that'll help them avoid praying mantises, right? It's just at this point in their evolutionary journey, this is the best model.
Or maybe they will speciate. Maybe there will be subspecies. Maybe they will specialize depending on how different competition niches work out and predation. And we don't know that, but they've gotten to where they are, which is for now good enough.
Yeah. And so I think the reason I was so interested by this story is that it combined all these different types of camouflage. They have coloration, they have physical mimicry, and they have locomotor mimicry. So they have they're moving in a particular way, they're morphing their body around to a particular shape, and they are trying to blend in with their environment. So camouflage mimicry on so many levels, it's very cool that they've been able to kind of stack all these things together.
It's amazing that they've been able to stack all. I mean, it's a lot.
Yeah.
I mean, the probability of all these various things coming together to allow them to survive and adapt to the conditions that they're in. That's amazing.
Please don't eat me.
Don't eat me. I would love to know, how widespread these spiders are, how big their populations are. Are these, are these spiders in a very specialized environment, habitat already? Are they in danger? Usually when you have very specialized organisms, they're going to be less likely to adapt to rapid change. So,
great question.
I wonder.
I don't have a range map for them. They're in a larger genus that's full of jumping spiders.
Lots of spiders that jump and they don't.
Yeah, and the majority of the genus specializes in hunting ants, which is interesting because this individual depends on them.
Well,
if they still eat them, they probably do.
Yeah, they look like them, and then they blend in, and then they eat them.
No.
I survived.
No one here but us ants.
Don't look at me. I'm just an ant. Just hanging out, just like you.
So cool.
I'm totally just like you. That's all. Oh my goodness. Wow. Are we all just ants mimicking what we can to survive? I don't know. Right? Putting on our colorful paint and moving our arms in various ways. I look like an ant. It's great. Don't eat me. Anyway, moving on to some brain stuff. Let's get into some brain things. We've talked previously on the show about something that is a very fascinating concept, and we don't really know why it's working the way that it is, but there's evidence that a certain frequency of light and also of sound, 40 hertz, which is also similar to the gamma wave brain signal within our brain, 40 hertz frequencies of light and sound have been shown to benefit Alzheimer's, dementia, other diseases of the brain related to aging. And so a group out of MIT said, well, let's look at tactile stimulation. And so they took a group of mice that are kind of predisposed to developing Alzheimer's neurodegeneration. It's the tau P301S mouse. It recapitulates the disease's tau pathology, where the tau tangles get into the brain and lead to a lot of the symptoms of behavioral change, memory loss, et cetera. And they they put the mouse cages on speakers. On top of bass speakers, actually, that were playing a 40 hertz sound. So it's a very low frequency sound in terms of it's within human hearing, but it's a low frequency. And the tactile vibration of the speakers vibrating actually led to changes in neural activity. The brains of these Alzheimer's mice were less likely to show tau tangle activity. They had increases in various components that are more neuroprotective rather than neurodegenerative. And mice stimulated for 3 weeks. I mean, could you imagine sitting on a bass speaker for like 3 weeks? They had preservation of neurons compared to mice that had not been put on speakers for 3 weeks.
So maybe not a speaker, but hear me out. What about prescription massage chair?
Right? Yeah. And do you need to be vibrated at 40 hertz consistently for 3 weeks? Or is this something that could be on and off?
Is it a scalable thing too? Because right. Is it auditory or manual? Like what is the impact coming from? Because there's the frequency of the vibration, which is physical, and then there's the frequency of the sound that is causing the vibration.
Yes.
Which is auditory.
Which is auditory. Yeah. So and we know that auditory 40 hertz stimulation can also be beneficial. So they've controlled for these things. It's just, it was the vibration.
So massage chair and headphones is what it would have to be.
And maybe a pair of special glasses that blink lights at you.
So basically I'm going to a sensory deprivation chamber for a while. I'll be
40 hertz spa.
But this is my other question. Does it matter that mice hear at different frequencies than humans?
And that is an interesting question.
And does it matter that they're smaller than us? So the physical vibration would feel different. Like, does it have to be scalable? Like, is it going to have to be a way crazy higher vibration for us? Or is it, I don't know.
Right? So many questions. I don't know. And I think it's, this is the beginning of looking into this fascinating direction for therapeutic, non-invasive treatment, right?
So we need a scaled animal model. We need something larger.
Yeah. And is it something that's really going to, it's not going to prevent the disease in the long run, but just if it can ameliorate deterioration, slow things down, help things out for just a little while, right? It could be, and yes, this is just mice at this point in time. So we don't know how this will work on people. I mean, there are people who talk about, the harmonics or the frequencies that are involved in cat purring being therapeutic. So I don't, that's not on your brain. I mean, you put on your cat hat for a while, let my cat sleep on my head.
As long as you don't spook them.
Yeah. And then you have claws in your
Lose
cranium.
an eyeball.
Yeah, that's no good. I don't like it at all. But it's just, yeah, it's a fascinating story that I'm surprised to see it continue in this manner, and I'm going to be very interested to see where it goes. Because first it was like, oh, we put this blue light at this, this weird
Yeah.
40 frequency light, the sound, but I was like, what is going on? And now it's a different modality, and if it can help ameliorate pathology, help behaviors, I mean, 40 hertz, it couldn't hurt.
That's one of those things when you go to like an alternative health spa and they ring a bell and they're like, this is going to heal you. And you're like, no, it's not. But it might. There might be some science behind it. Who knows?
Right. So that's like where a lot of these things, they start blending a bit. And it's like, okay, there is probably some truth to some things, but we just haven't studied it and figured out the mechanisms, and we don't understand exactly why. And some of these things are absolute, bullocks. But it's just a matter of figuring out.
Some aren't. Also, like, where did it come from? Was this something that humans did for thousands of years and we stopped doing it and it was working, and then we stopped because it was like, what is this? But then they did it for thousands of years because there was a reason it worked. Let's find out scientifically why, versus I've decided that if I carry this crystal around, I won't get cancer. That's very different than something that's based in kind of more ancient wisdom that might have a reason for having worked in the past. So
I don't know. I don't know. I like reason. That's why I'm here. We can't ever assume that we know everything. I think what is interesting about the 40 hertz signal is that it is the gamma wave so it is a brain wave that is part of our brain signaling. And so that in itself is interesting. Why is that one working? What's the mechanism there? But beyond vibrating 40 hertz, researchers at the Reinhardt Lab in Boston University have looked into transcranial alternating current stimulation. So last week I talked a bit about like using magnets on your brain to treat depression. This week, it's not magnets, it's just alternating current on your head. So like if you have ever used like a TINS, a muscle stimulator to get alternating current to stimulate your muscles, maybe can help with muscle cramps or muscles strengthening. You can also, people have been using them at particular frequencies and in particular locations on their heads to stimulate neural activity within their brains. And it has been shown over time, positive and negative results. I don't know, lots of studies going different directions. So they did a meta study, a meta review of a number of 100 over 100 published studies, total of over 2,800 human participants, to look and see whether or not this transcranial alternating current stimulation has an impact on cognitive functions, whether it's only for people who are experiencing mental deterioration or mentally challenged in any way, or whether it helps healthy people as well. And really, they found it just works. It helps everybody. It's not just helping people who already have disabilities or deficiencies. It's also, it can also enhance cognitive function. It does depend on where the electrodes are placed. They found that a specialized type of this tax can target two brain regions at the same time, manipulating how they communicate with each other. And so that in itself can either enhance or reduce cognitive function because it's affecting how the brain is communicating within itself. But it's definitely a technique that has promise. So yeah, stimulating your brain with electricity.
And this might be a really silly question, but how is this different from the electroshock therapy of like the 70s and 80s?
So the difference is that the electroshock therapy was much like, it's higher voltage. The electroshock therapy was more focused on the frontal lobe. The TAX stimulation is a lot lower voltage. It's different time frames. So it's like the amount of time that the stimulation is occurring is different. And then also um, where the electrodes are placed is also very impactful.
Okay.
So there are actually
the electroshock therapy was like a blunt object kind of.
like a sledgehammer.
Yeah.
And now this is like, oh, we're getting into needlework.
Okay, got it. Okay. Thank you. I appreciate the clarification because I was like, I thought we did this and it mixed results.
Right. So yes, mixed results
Okay.
on the sledgehammer effect, right? And mixed
Yeah.
results also with this needlework as we get more and more refined and the resolution is, increased and we start getting more specific into what areas of the brain are impacted. But there appears to actually be a moderate improvement, sometimes great improvement, depending on what's happening and who it's happening to and what area of the brain is involved related to working memory, long-term memory, attention, executive control, intelligence, how you interact with the world around you. So, yeah. Yes.
Well, I'm not ready to electrify my brain, but
it's not for everyone. It's not for most people, probably. But it's interesting. It's probably more so going to be used in a therapeutic nature. And that's probably where it's going to be seeing the most promise moving into the future. I did want to bring up a couple of cool studies that I didn't put into the notes, or actually one specific one that people are talking about this week that at first I had been like, oh, whatever, spinal implant, stimulating the spine, helping people walk, blah, blah,
I did see that.
But this is really cool what they're doing. Okay, so researchers have published in Nature this week their work with one individual who's part of a study and how they have created a system that takes the intent of movement from the brain. So they've got cranial implants, cortical implants that are positioned to read the electrical activity that reads the intent of the individual who was injured, had issues, had kind of hit a plateau with walking with a walker and hadn't been able to get to a point where he was able to completely return to normal movement or walking activity. They put these cortical implants in. They have a backpack, which is like they call it a wearable processing unit that receives that data from the brain and then basically as an AI and sends stimulation through an implantable pulse generator and electrodes that are in the spine below the level of the injury to move muscles in the legs. And where previously he was unable to move his hip flexor muscles within a very short period of time after just first getting this system put on, getting over the implantation and getting it moving without a lot of training, he was able to start thinking about moving his hip flexors, and his hip flexors were moving. He's recovered a lot of his independent activity, able to walk without a walker, is able to move, still there's a lot of difficulty involved, but in the years that he has been using this, it has led to recovery that is beyond just using the stimulator. So his body is starting to repair and fix some of those connections that were missing.
How long ago did he get this?
It's several years ago, so it's about
Wow,
it's they in the discussion. I think they say it's something like over 3 years or so.
that's
But
amazing. Well, and I love that it's already a small enough processor that it's a backpack, and
right,
if this continues and they continue to develop it, it'll be a bracelet, and then it'll be a necklace, and then it'll be like a lapel pin, and you'll be like good to go, right? It's going to keep
I'm
getting
good to go.
smaller and
Or it's completely implantable, right?
mhm.
You don't need
Yeah,
a backpack. You've just got something that's just a tattoo in your skin, or just under the
right.
skin.
Yeah,
Yeah,
just inject a little pill shaped thing, and you're good to go. This I love this.
it's amazing.
This is a really smart connection of a bunch of different kind of science that's been happening all at once. This is the perfect concert of bringing them together for a real improvement in somebody's life. And I love that this isn't even just we tried it and it worked. It's this person's life has been improved for over
Yes,
several years.
and it's getting
That's
and
amazing.
his autonomy is improving, increasing. Life is getting better. Who knows if the lesion in his spinal cord will ever be completely healed, or get to that point. But this is an amazing part of that. And there's definitely, within the nervous system, we know there's this mantra in neuroscience is use it or lose it. And there's
Yeah.
also feedback loops. So if you are unable to use something, there is atrophy. But if
Right.
you're able to use it, then the feedback loop becomes positive then. And so maybe that will help the healing.
Yeah, absolutely. That makes nothing but sense. Is you get to keep the muscles moving. You're not to mention you might be building new pathways in your brain to figure out how to get around certain issues that are preventing you from being able to ambulate normally. It's
Oh my goodness, I know.
it's very cool.
Yeah. So I wasn't originally going to talk about this story, but I took a deeper look into the study, which is available through Nature, and it's an incredible advance. All the stuff that they're doing, and like you said, where is it going to go from here? How many people is this going to work for? I mean, I guess the question is also, who's going to be at the place where they're like, yes, put things in my head, you know, you know, cut a hole in in my skull and put implants in my brain and make it work.
Well,
So
not everybody who has hearing loss or has been born without hearing wants a cochlear implant, but like
right.
having them available is
Yeah.
still a huge improvement on quality of life for people who want to have that procedure. So it's
Yep.
you're right. It's not everybody's going to want it, but particularly if somebody gets injured later in life and has a harder time adjusting to a new lifestyle, I think it might be very attractive.
And it's just amazing to me that we know enough at this point in time to be able to put the electrodes in the correct place in the spinal cord and just be able to go, okay, your brain thought this. We're just going to send that information to the
Yes.
spinal cord right now and make that work.
I love it.
Here you
Oh, man.
go.
So cool.
Yeah.
Next stop, robot legs, right?
Yeah. And so this, additionally, this is not just the paper in Nature is walking naturally after spinal cord injury using a brain spine interface. There's also, robot legs, brain spine interface. We can add more legs, or maybe we can add superpowers to your legs,
Oh my
or
god. Well, actually, this is just basically the Iron Man suit, right? Like it's just, it is
except
it
it doesn't have the nuclear
no, no. I
power,
mean it
and
is and it isn't. I guess it's more it's sorry to attach it to the appropriate comic book thing. I guess it is
right,
more like Dr. Octavius's extra arms. That's more what it's
more
like
like it.
because
Yeah,
that's it is spine and it's taking his thoughts and moving the arms. So it's more similar to that. But
yeah.
it's
And
I
it's
don't
I mean, they put a thing in the brain, but then it's wireless and then the backpack, and then they're like, "Oh, electrodes in the spine," and I don't know. This whole thing
but
is
yeah, that means if you had limb
confusing.
loss and you could take brain waves and then you just need to create. It's very close to having a real-time response from artificial limbs as well. I think it's parallel, but I think it's going to help inform that kind of research as well.
I mean, just not just recovery from paralyzation or spinal injury, but maybe this will help additionally with limb loss. Maybe it will, like you said, maybe this is going to be, maybe we can take it further. It's just, the plasticity of the brain is, it's extreme. It can go places. It can take us places, which I think is cool. Oh, and like I mentioned, like very, very briefly, right before the show actually started, hypoxia. If you, Blair, want to inhale less oxygen, maybe it'll be good for you. Well, I mean, it's good for mice, possibly.
I'm not going to do it now because I have a passenger that I need to send oxygen to. So I'm gonna wait. I'm gonna wait to figure this out. But tell me about it.
Yep, publishing in PLOS Biology, researchers used a mouse model of accelerated aging, and they gave them less oxygen. So instead of caloric restriction, it was oxygen restriction, and oxygen restriction seemed to help their brains. They don't know why. The mechanisms are completely unknown, but oxygen restriction led to extended lifespan in these aging accelerated mice.
So then people in Denver should be living longer.
Yep.
And oxygen bars are bad for you, is what I'm hearing.
Yes, it's very interesting conclusions, or related ideas. But there is a suggestion from some data that there is less metabolic disease, diabetes, and cardiopulmonary vascular disease, COPD disease in higher altitudes, and maybe some of this is related to the oxygen levels. But I don't know. There are also other drawbacks, higher suicide rates. I don't know. Things are very. Don't go restricting your oxygen just yet, folks.
No thanks.
No,
wasn't that the whole thing about COVID that was like causing real lasting problems to people who had real bad COVID?
Right.
Was oxygen deprivation. So I'm good.
Yeah. Just stick with your oxygen the way that it's given to you. And, I don't know. I'm going to wait for that 40 hertz spa. It won't hurt. Have we done it?
We've done it.
Have we come to the end of our stories?
Mm-hmm.
We did it. All right.
Ooh, I got the hiccups just in time.
It was perfectly timed. Everyone, thank you so much for joining us for another episode of This Week in Science. We hope that you enjoyed the show. I need to thank very special people for all of their help. Thank you for everyone in the chat rooms, for being here in the chat rooms, keeping up with us while we were talking about things and making comments and giving us your thoughts. We really appreciate you in the chat. Additionally, Fada, thank you so much for all of your work on social media and show notes. Identity 4, thank you for recording the show. Gord, Arnlore, others, thank you for helping make those chat rooms happy places to be. And Rachel, thank you for editing the show. Additionally, cannot go on without specifically saying thank you to our Patreon sponsors. Thank you to those of you who make TWIS possible. Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstead, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence, Lamb, John McKee, Greg Riley, Mark Hassenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rapin, Richard, Brendan Minish, Johnny Gridley, Clemy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Caldery, Marjorie, Paul D. Disney, David Summerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon and we didn't read your name here, head over to twist.org and click on the Patreon link and go from there. On next week's show, we will be back on Wednesday, 8 p.m. Pacific time, broadcasting live from Twitch, YouTube, and Facebook. And you can find more information at twist.org/live.
Want to listen to us as a podcast? Perhaps while you sit on a giant speaker and feel the vibrations. Search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
And for more information on anything you've heard here today, show notes and links to stories are going to be available, like I said, at our website, twist.org, and you can sign up for our newsletter.
You can also contact us directly. Email Kiki at Kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put twist, TWIS, in the subject line, or your email will be spam filtered into the rings of Saturn, which we all know will dissipate over the next 100 million years or so. So we're not going to get to it in that amount of time. So just
Have you seen my inbox? Yeah, no, I'm not going to get to it. You can also ping us on the Twitters where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. And when we say Twitter, we also mean things like Instagram and Twitch and Facebook and all other places. We love your feedback. If there's a topic for a subject or anything you want us to cover or address, suggestion for an interview, let us know.
We'll be back here next week, and we hope you'll join us again for more great science news. And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.